All right, I think we can go ahead and get started. Hi everyone. Thank you so much for joining our webinar today on tips and best practices for isolating synaptosomes. Today, our speaker will be Dr. Sandeep Rajkumar, a postdoctoral fellow at the University of California, San Diego. My name is Afrida Rahman Enyer, and I'm going to be your host for today. But before I go ahead and pass it off to Dr. Rajkumar, I just want to share a few introductory slides about Protein Tech in case you are unaware of who we are as a company. So Protein Tech is a leading manufacturer of antibodies and immunoassays. We have a really expansive portfolio of antibodies that encompass many different research areas. We also offer ELISA kits and IHC kits, as well as cell separation kits and antibody labeling kits. Within our Protein Tech brand, we also have our humankind branded cytokines and growth factors. All our proteins are expressed in human HEC293 cells, which means that you can be confident that they have the native folding, post-translational modifications, and glycosylation that you would expect to see in human proteins. And that in turn gives them greater bioactivity. We also have our Chromatec branded nanobody based reagents. Nanobodies are about 10 times smaller than traditional antibodies, and they bind to their targets with really high affinity, making them ideal for immunoprecipitation, super resolution microscopy, and protein purification. And lastly, we also have a subsidiary of Protein Tech known as Protein Tech Genomics. And Protein Tech Genomics manufactures antibodies that are conjugated to oligonucleotides for single cell RNA sequencing. As a company that is driven by scientists, we're here to support our researchers and scientific community any way that we can. So, our award winning products are self manufactured, and we offer the largest in house portfolio of antibodies for the human proteome. We also know the importance of product validation, and we offer open access validation data. When testing new products, we do validate with relevant sample types, appropriate experimental conditions, and we do use knockout and knockdown models whenever possible. Also, as antibody experts, we do have in-depth knowledge of immunoassay applications, and we can offer outstanding technical support for our customers. We also stay on top with cutting edge technology, so such as our proprietary antigen specific B cell cloning and engineering platform, which is what we use to manufacture our UniRab recombinant antibodies. With 7 inventory sites and manufacturing and research sites on 3 continents, we are able to offer next day delivery to many areas in the world so that you can get started on your experiments right away. So as a company, we do measure our success by our customers' scientific success, specifically by helping them publish impactful research faster. And so this graph here on the left shows that over time, over our company history, our products are featured in more and more publications each year. And as of right now, we have over 280,000 publication citations, making us one of the top 5 cited antibody companies and also one of the fastest growing companies in product citations. So with that, I would like to go ahead and introduce our speaker for today, Dr. Sandeep Rajkumar. So Sandeep is currently a postdoctoral research in the Department of Psychiatry at UCSD. He did his PhD in neuroscience from University in Germany on the characterization of various cellular and synaptic phenotypes of ALS using iPSC-derived motor neurons and cerebral and spinal organoids. During his time at University, Sandeep and his colleagues published their method for synaptosome isolation from hiPSC motor neurons in STAR protocols by Cell Press. Sandeep then went on to do a postdoc at Boston University in the Department of Anatomy and Neurobiology, where he focused on evaluating molecular and functional readouts of Down syndrome using CRISPR edited iPSC derived cortical organoids and forebrain assembloids. Sandeep has experience in various molecular techniques such as immunostaining, Western blotting, and PCR, along with multi-electrode array, clarity, confocal microscopy, 3D live imaging, and calcium imaging. So it's really great to have his expertise here today and to hear what he has to say about synaptosome isolation. So thank you so much for being here, and I'll go ahead and pass it off to you, Sandeep.
Yeah. Thank you very much, Afrida. Let me just share my screen. Okay, so like Afrida already mentioned, so currently I'm doing my postdoc research at the University of California, San Diego. But this presentation is going to be about a technique about synaptosome isolation from iPSC-derived motor neurons, which me and my colleagues did at the Ulm University, where I did my PhD. So we published a protocol paper in early 2023, and today I'll be explaining a bit about synaptosomes, what they are, why we need them, how to isolate them, what have been some techniques that have been used in the past decades, and how we try to modify it and adapt it for iPSC-derived neurons. And I'll go step by step and try to explain how to isolate them. And in the end, we'll discuss a bit about the applications of them. So just to start off with, I'll give a brief introduction about a synapses. So I'm not going to go into detail about what are neurons and what synapses do. And I'm assuming most people will have this basic knowledge. But the most important thing when it comes to synaptosomes is what's inside a synapse. So in the synapse, obviously, there's going to be a presynaptic compartment and a postsynaptic compartment. So in the presynapse, you're going to have your synaptic vesicles that package and release your neurotransmitters, which is obviously important in generating action potentials. And when they bind to the membrane, they form these active zones, which have dense projections. And here you have protein complexes like piccolo and bassoon, and you have lots of voltage-gated calcium channels, you have cell adhesion molecules, and you also have a mitochondria present in these compartments. And in the post-synapse, you have your neurotransmitter receptors like ion channels, and you have scaffolding proteins like HOMR1 and your various shank proteins. And all of these are found on this very electron-dense region known as the postsynaptic density, which is a very thick membrane region. And this is important later because in synaptosomes, we're going to be isolating these compartments individually. Apart from these, as the synapses, you also have RNA and microRNAs, and lots of older techniques have been able to isolate these from the synapses as well. And why do we need to study synapses? So synapses, apart from being the integral component of physiology in neuroscience, where they needed to generate action potential, they're involved in learning and memory through plasticity. They also have a role in disease pathology. So this is what we were focusing on during my PhD. So various neurological conditions like epilepsy, you have neurodegeneration like Alzheimer's, ALS, Parkinson's disease, neurodevelopmental disorders like autism spectrum disorders and Down syndrome, and psychiatric disorders like schizophrenia, depression, and other substance abuse disorders. And all of these, there are some changes happening at the synapses. It could be differences in protein expression. It could be differences in neurotransmitter release, reuptake. It could be structural changes. So it is important to study a synapse, and this is why we need synaptosomes. So what are synaptosomes? So synaptosomes are basically you isolate the presynaptic terminal that contains your vesicles and mitochondria and all the other stuff, and your postsynaptic membrane with the enriched postsynaptic density with the ion channels and the scaffolding proteins, and you isolate them from the neurons. So if you have a brain tissue using, I will explain a bit later about the steps. So using a different steps, you will be able to isolate just the presynapse and the postsynapse, and you form these structures called synaptosomes. And why do we need to study synaptosomes? So the first important thing we can do with synaptosomes is, let's say you have a protein of interest and you don't know its function, but you hypothesize that it could be involved in synapses, and you don't know if it's going to be presynaptic or postsynaptic. And then in this case, you isolate the synaptosomes, you isolate its subcompartments, and you do a simple Western blot, for instance, and you'll be able to find out in which compartment your protein of interest is. Another thing you can do is you can quantify the expression of different synaptic proteins in your condition. So you can have healthy versus diseased, or you can do a drug treatment and see what happens later. And you can quantify how the expression of this protein changes in different synaptic subcompartments. And there have been lots of protocols where they look at not just the protein expression, but also mRNA and also microRNA expression from various synaptosomal techniques. So in a way, synaptosomes, they offer like a snapshot view, like a microscopic view of what is happening within the synapses. And you learn lots of things like expression of proteins and localization of RNA and also different other proteins. So I'm just going to go give a brief history lesson, let's say, about how synaptosomes came to be. This was back in the 50s when people were still trying to identify different neurotransmitters. At that point, acetylcholine was well known as a neurotransmitter, but at that point, people thought it was found in mitochondria, for instance, because it was involved in energetics. and they didn't know that was found in this the synapses. So there was a protocol back in the 50s and the 60s where by using multiple centrifugation steps and multiple density gradients using sucrose, people were able to isolate different fractions or subfractions of brain tissue. In this case, they used dog brains. And then they found acetylcholine was found in what they called the nerve ending. So there was higher expression of this there instead of in the mitochondria fraction. And that's when they realized that neurotransmitters are probably found in a different region. And indeed, in the summary of that paper, they say that this neurotransmitter is not in the mitochondria, but in another type of organelle. So they didn't even know that it was in a status. But later in the 70s and the 80s, there were more methods where they isolated different parts of the synaptosome. So once again, the steps would include you homogenize your brain tissue. In this case, you do multiple centrifugations in a sucrose density. And in the end, you will get something like this. You have the lighter membranes, you have myelin a layer, and then you have your synaptosomes here, and then you have mitochondria being pelleted. And then you'll have to carefully aspirate these different layers depending on your research question. And then you can just solubilize them in buffers, and then you go ahead and you can isolate the postsynaptic density fraction. So in the end, you'll get something like this at the pellet. And in your synaptosomes will contain your presynaptic and your postsynaptic regions. And then you further separate them into presynaptic and your postsynaptic density fraction. So these were the traditional protocols that have been in use for many decades, where you need to have a gradient involving sucrose. You can use phycol or percol and try to get different layers. And this protocol has been used, like I said, for over decades. There are lots of pros and cons associated with it. So the pros are obviously you get so many different subcompartments, and you can further purify them and you can separate them. So this is going to be useful in lots of areas, and it's highly specific. So as long as you aspirate those layers out of the sucrose gradient, these are highly specific. But the cons are that you need a large quantity of tissue. So for instance, if you're going to use rat brains or mouse brains, for instance, you need at least 10 to 15 cortexes from 10 to 15 animals to get enough material for this technique. And if you're going to go for a smaller structure like a hippocampus, you'll need hippocampi from 20 to 25 mouse. And then this is going to do, obviously, you're going to use up a lot of animals just for this. And there's going to be difficulties in scaling it up or down. You need large quantities of buffers. You need larger tubes for the sucrose gradient, and then you will probably need larger centrifuges as well. And another problem is that it takes longer. It takes about 4 hours to get these gradients properly. So what happened in our lab from early 2010s, I would say, this is before I joined the lab, obviously, they developed a faster protocol. And this protocol does not use a gradient. So basically, you just use multiple centrifugation steps and with different buffers, and then you're able to separate some of the layers. You won't be able to get layers like myelin and mitochondria separately, but you will be able to get synaptosomes and later separate them into a synaptic cytosol and postsynaptic density. And it can be scaled up and down, and you require lower quantity of tissue. So you just need, I don't know, 10 to 15 milligrams of tissue in the end. So that's not much. And this can be done in approximately 2 hours. So this is just an overview of the protocol that was adapted for so I will go in detail about each of the steps in the later slides, but just to give an overview, first step is obviously you homogenize your tissue, then you prepare a lysate and you purify it so that you remove cell debris, extracellular matrix, and all of those. And then you centrifuge it and you'll get the cytosol and then the crude synaptosome separately. And then by using Triton X100 solubility in the cytosomes, in the synaptosomes, what happens is the cytosol is solubilized, and then the pellet that contains the insoluble fraction will have your enriched postsynaptic density. So it's basically just a 4-step protocol, and it could be done in less than 2 hours. So using this protocol in the lab, they used it to study autism spectrum disorders. Like there was a study published in the lab where they studied differences in dendritic spine dynamics, looking at proteins like porphyrin and drebrin. And there's also in mouse models of autism spectrum disorders from regions like the hippocampus and the striatum, where they were able to separate different fractions and look at the expression of various synaptic proteins. However, at this point, around 2022, 2023, we were not able to find a protocol for culture neurons. So until then, all the protocols were for brain tissue from rodents or from postmortem humans. And so we decided to adapt this shorter, faster protocol for iPSC motor neuron synaptosome isolation, because that was my PhD project. So we were looking at ALS research. So we wanted to adapt this for iPSC motor neurons because we were seeing difference in synaptic activity and protein expression just by immunostaining and Western blotting. So we wanted to look into it in much more detail using synaptosomes. So now I'll explain each of the steps in a bit more detail. And this is the paper that we published. This is the protocol paper. And initially, of course, we cultured our iPSC-derived motor neurons in 6-well plates. So we were seeding around 30,000 iPSCs per well, and they would grow into motor neurons. And we kept the number consistent across our ALS motor neurons and our healthy motor neurons. And then the initial step is to prepare a homogenate. So this is similar to how we would prepare samples for your Western blot, for instance. You just need to have a buffer. In this case, this is the buffer that we use. And you just add 1 ml and you homogenize it with a tissue grinder. You can use like a mortar and pestle or whichever way you want. And then you save some of it so that you can use it for Western blot or proteomics later. And the rest of it, you're going to use it for the next step. For the next step, you just centrifuge it for about 5 minutes at 500 G. You will get a pellet, which contains the cell debris, nuclei, and ECM. So if you need it, you can use it. Otherwise, you could discard it. And a supernatant will contain all your neurons. So this is the cell bodies with the cytosol and the membranes, all of it. So we're calling it the S1 fraction. So we give it like different notations, like homogenate is HO, and this fraction, the supernatant is called S1. You can save a bit of it, like 100 microliters for later steps, like blotting again. And the rest of it goes into the next steps. So in the next step, what we do is we do another centrifugation, this time for 15 minutes, around 10,000 G. And once again, you get a supernatant and your pellet. And now in your supernatant, you will have your cytosols. So all your cytoplasmic proteins will be in this. We're going to call this S2. There will be some membrane proteins attached because this is a crude centrifugation step. So it won't be a complete separation or purification, unlike the sucrose layers that you saw in the earlier slides. And then in the pellet, you will have what we call a crude synaptosomal pellet. So this will have your pre and post-synaptic compartments. It will have your cytosol, it will have your vesicles, it will have your post-synaptic density with all your receptors and your scaffolding proteins, all of them here. So if we were to do a Western blot and compare the different fractions so far, so this is part of a larger Western blot, you will see that there is not much separation between the S1 and S2. So the separation happens in one of the later steps. So I'll show you next. So the next step is to use a buffer that contains Triton X100 in order to solubilize your cytosol, and then your postsynaptic density will be insoluble. So for that, we will add this buffer. We will centrifuge it for 80 minutes at 20,000 G, and then you will have a supernatant with your soluble synaptic cytosol fraction, and then you will have a pellet which has your synaptic membrane. So this has some membranes are from the presynaptic compartment, and mostly you will have your postsynaptic density. And then all you have to do is just solubilize this fraction using a buffer that contains sodium deoxycholate. And if you want to know what happens in these buffers, you can look at the paper that I showed earlier. So it's explained in a bit more detail. So now you will have multiple fractions. Each will have different kinds of proteins. And then all of this is done in 2 hours. And then you will have a complete Western blot like looks like this. So in this case, we are using cholineacetyl transferase, which is a motor neuron marker, which is found in the cytosol. As you can see, you will see its expression in your cytosolic fractions, but you barely see it in your synaptic fraction. And then the next protein is HOMER1, which is a postsynaptic protein. And you'll see that its expression is not as much, or it's more diluted in your earlier fraction, but it's significantly enriched in your postsynaptic density fraction where it's supposed to be. And then actin is a commonly used internal control in Western blots, and here we're using it as a cytoskeletal marker. And you see that it's barely present in the synaptic cytosol fraction, but it's enriched in your membrane fraction, where obviously you have scaffolding proteins and you have cytoskeleton attached to that as well. Then synaptophysin is a presynaptic protein, and you see that its expression is robust in some of the earlier fractions, and it's highly enriched in your synaptic fraction because that's where it is, because it's bound to synaptic vesicles, and it's barely present in your postsynaptic density fraction. So this shows that the separation was there, and then the proteins are expressed where you want them to be expressed there in their specific fractions. And this is like more or less the whole protocol. So it takes in less than 2 hours. You just need enough starting material of neurons, and then you just do multiple centrifugations, and then you get all of this. And then you can also quantify these Western blots. You can run other proteins as well. You can run all the other fractions as well. And then you can just check what is the enrichment percentage. You just need to see that they're higher than a one-fold enrichment. Then you confirm that your protein is present in your subcompartment of your interest. For instance, synaptophysin, it is heavily enriched here in your S3 fraction, where you have the synaptic cytosol. Your Homer one, which is postsynaptic, is heavily enriched in your P3 fraction, which contains obviously the postsynaptic density. So it's not just by Western blot. We also have done proteomics on these samples just to confirm. And we saw that these synaptosomal fractions are heavily enriched with terms for synaptic proteins. So this shows that our method was efficient, it was successful, and it is good enough for high throughput techniques like proteomics. And like I said, so there haven't been protocols for 2D cultures and iPSC neurons so far, so we were able to use this as well. And like I mentioned earlier, you can use this for your major applications. You can check your protein of interest to see in which fraction it's there, so that could be useful. You can use quantitative analysis. You can like measure the expression of these proteins across different fractions. You can use it in proteomics. You can do what is known as a synaptomics, where you do proteomics of just the synaptosomes, and you see the expression of various proteins and across your groups, whatever your test groups are. And we use these synaptosomal techniques in our ALS research, and we published a paper once again in 2023, where we took iPSC-derived motor neurons from 4 different ALS mutations, so 2 lines each, and 4 healthy controls. And we did, once again, proteomics with these synaptosomes to see what's the difference between healthy and ALS motor neurons. And we saw significant downregulation of presynaptic vesicle proteins that are involved, obviously, in neurotransmitter release and vesicle-mediated transport, so in docking and release of neurotransmitters. So we did see what we were seeing earlier based on just immunofluorescence or Western blot. We saw it much more in detail using synaptosomal proteomics, that there was indeed a dysfunction of presynaptic proteins in ALS. So this is one of the applications that we mentioned in disease research, and this can be obviously applied to various other diseases. Like you can check it in autism spectrum disorders, you can check it in Alzheimer's and other disorders as well. So obviously there are certain limitations, and there are ways to overcome them as well. Like I mentioned earlier, this is not a 100% pure separation. There will be some, let's say, contamination from other proteins in the various ones. But still, if you see enough enrichment in the various fractions, I think this protocol is good enough, and it has been validated by proteomics. And so I would say this protocol it offers like a balance between you don't get 100% pure separation, but it's still very, very good enough to be validated by proteomics, and it can be used in disease research as well. The second thing is, when you're using this for in vitro cultures, different people use different protocols, and they also use different types of neurons, like hippocampal neurons or cortical neurons. For each of them, the maturation time points vary, obviously. So when you use this technique in a time point where you don't have synapses anymore, whether they're dead or they haven't developed yet, then obviously you won't see any signal when you do your Western blots or you confirm them. So it's important to identify which time point is important for you and whether your neuron cultures are mature in vitro. And then this can be determined. You can use immunofluorescence, you can use electrophysiology or electron microscopy, and you can see if your neuron cultures are mature enough, and then you can use this technique. Another thing is that you definitely need an internal control, and it needs to be a protein that does not change too much within all your fractions and across your control and your disease groups or your treated groups. And for this, you'll probably need to check, because in some conditions, even actin could be changed if you're going to use that as a internal control. So it is important to check that you have a good internal control that does not change much across fractions. And in proteomics, of course, you can look at the fold change, and you can choose proteins that don't change, and you can use this as an internal control, and you can standardize your proteins to that. The thing is, like I mentioned earlier, you can also adapt this technique for different types of neuron culture, and you obviously need to standardize them. You may have to play with the buffer volumes a bit, and you can also adapt it for different models. I had mentioned earlier that this model was initially developed for rat and mouse brains, and we adapted it for iPSC neurons, but you can use it for different other models as well. You just need to scale up and down the buffer volumes based on the tissue amount that you have or the number of neurons you have in culture and their maturity. One example is this protocol was adapted recently for Drosophila brains. So in this case, so this means that the model, that the technique is valid and you can use it across multiple models. But ideally, we wanted to have a protocol for iPSC-derived neurons, and hopefully this can be useful. I'll have to note that we have not tried to extract RNA or microRNA from this, but I know that there are other research labs that are trying to do it, and hopefully they'll be successful. Obviously, you need to use it, RNase inhibitors in your buffers and maybe have uh, different concentration of certain components so that your RNA does not get degraded. But I think it is possible to isolate RNA from these. So with this, I hope everyone had an overview of how to isolate synaptosomes from iPSC neurons, what are things that can be done with this, how good it is, and what are the things that are limitations, but they can be overcome as well. So with this, I would like to thank the people at Ulm University who have made this technique, and also thanks to Protein Tech for giving me this opportunity for this presentation. And thank you for everyone present here for your attention. And I'm open for questions.
Thank you so much for that really very informative talk. I learned quite a bit. We have quite a few questions rolling in, so hopefully we will get to most of them. If we do not get to your question live at the very end, I do have Sandeep's email on a slide, so you can email and reach out to him directly, or you can email us and we can get him the questions as well. But we'll go ahead and get started. There were quite a few questions asking about starting material and how much of the starting material you suggest for this method.
Yeah, so in that case, we would seed iPSCs into these 6-well plates. So all 6 wells will have 30,000 iPSCs per well. And obviously, these will grow and they will become a motor neuron. So this was standardized for us when we were doing Western blot experiments, and we decided to continue that for this experiment as well. And this needs to be standardized for people's protocols. So obviously, if you're going to use a different protocol for cortical neurons, for instance, or hippocampal neurons, maybe they grow differently, maybe they mature differently. So you will need to standardize this as well. So my suggestion would be to just do a test run. You can do like a couple of 2 or 3 plates. And one you have more neurons, and one you have less neurons, and then you try to isolate the synaptosomes. And all you just need to see is your specific protein needs to be in its specific subcompartment, and it needs to be enriched. So it should be at least 1.5 or 2 times, twofold higher than what is in your original, the S1 fraction or your homogeneous. So that means it is enriched. So that's the only
And this
consideration.
method,
So it needs
there
to
was
be.
another question about
Mhm.
kind of tissue versus cultures. So this method could work for both tissue and cultures.
Yeah. So this was initially developed for rodent brains, for mouse brains and rat brains, and we adapted it for iPSC neurons by scaling down the buffer volumes. And obviously, you can scale it up, scale it down, depending on the amount of tissue you have, what kind of tissue. If you're going to use post-mortem human brains, for instance, you could have lots of material, and then you probably need to use more buffer as well. So that needs to be standardized, and it's going to be different with different people. And it's also dependent on your research question as well, which fractions you need. If you need further purification, you can also stop at earlier steps. If you just need the synaptosome and you're not interested in the different fractions. So it, once again, depends on different researchers.
Great, thanks. And then there was also a question asking about what is the average protein amount that is isolated?
Oh, yeah. So this we have it in our protocol paper. I will need to check because in that one we did a validation of this Western blot that we did, and we also estimated the concentrations of each of these. I think you will get good enough concentration to do at least 4 or 5 Western blots. I mean, and then we also had enough material to do proteomics. So I don't know the exact number. I'll have to check it, or you can check the publication that we have. And you can also check this publication that we have where we used multiple cell lines. So we had enough material. So it was for even for a smaller volume of about 70 microliter for this last fraction, we still had enough to do 4 or 5 Western blots and do proteomics. So I would say it is considerable.
Great, thanks. We also had a question asking, can a tabletop centrifuge be used for the isolation?
Yes, you can use a tabletop. You can use a fixed rotor centrifuge for the early steps. You have to probably look at the publication again for the last step. I think you will need a swing bucket centrifuge. but it's a tabletop. You don't need a big ultracentrifuge. I think a couple of them have a cooling step where you need to do them at 4 degrees Celsius. But apart from that, you just need a cooling centrifuge and you can use a small. So the first two steps, you used a small tabletop centrifuge for these, and only in the last step, we needed a larger one.
Great. Thank you. We also had a question regarding doing Western blot following the isolation. So what would be a good loading control for the Western blots done with just the synaptosome lysate loaded?
Yeah. So in our case, for instance, here, this Western blot has multiple fractions. And in this, we used actin as an internal control. So here you cannot really see actin in your S3 fraction. That's because we have intense signal coming from the actin from this fraction. But if you were to load only the S3 fractions from, let's say, all your groups, you will see actin there. And in our case, actin was not changed between our healthy and ALS group. So we use this as an internal control. And for the proteomics experiments, we had a set of proteins that were unchanged. And you will have the data sets probably in this paper published online. So you can use that. And then we had a set of proteins that were unchanged and we use that as internal controls. The important thing is you need to normalize it to the internal control in each fraction. And then you also have to normalize it to your total protein as well. That is found in one of these fractions. So probably your S1 fraction. So you need to do a double normalization if you're going to use Western blots. And in proteomics, like I mentioned, you can use whatever set of proteins are unchanged across your groups and across all the fractions.
Interesting. And kind of a follow-up question to that, have you ever looked at, isolation from different ages of animals and kind of compared those? Because I know with some of the standard housekeeping genes, the expression can depend based off of age.
Yeah. So I think in these couple of papers that they used, I think in one of them, I think in this one, there were multiple time points and multiple brain regions as well. And I think here I'll have to check what was the actual internal control. But yeah, so I think even in this case, actin was not changed. But yes, there are certain disease models where you will have changes in even your internal control over time because there is maturation. So it is important to normalize them internally first, like I mentioned earlier, and then you check the differences in your protein expression. And yeah, so it would vary. So you could do, I don't know, if tubulin is a better option, and there are other internal controls that are people are using different kinds of internal controls for different studies. So you'll have to check how it changes in your disease type and the maturation point of your neurons or your animal model.
Great, thank you. The next question is it possible to isolate synaptosomes from neuron-derived iPSCs without disrupting the membrane with Triton?
I think it depends what your research question is. So, for instance, if you were to stop at this point where we don't have the Triton in the buffer, you will have your synaptosome. It's just you will not have your cytosol and your postsynaptic density separated, but this fraction P2 will be like a combination of these, and it will look something like this. I don't have that plot here, but it would have both your synaptic cytosol and your postsynaptic density here, and you don't have to use Triton X. But the entire principle of separating the synaptic cytosol and the postsynaptic density has been with Triton X traditionally. So, for instance, even in the earlier protocols where you have to use sucrose gradients, the Triton X100 solubility is the main defining factor of separating the postsynaptic density and enrichment because it is insoluble at Triton X. So, if you want to have another compound or another chemical that does not disrupt it, I'm not aware of it, or it depends on your research question. You can stop here already, and then you don't have to use Triton X, and then your synaptosome is going to be intact. And I know that in some of the earlier protocols, people do electron microscopy on these fractions, and then you see the membranes like together like this, and you have your presynaptic and your postsynaptic density together, and you have a synaptosome. So that could be an option.
Great. That actually answers another question that I just saw where they asked if you've checked the synaptosomal fraction identity with electron microscopy, but it seems like you can do that. I don't know if you have specifically done it yourself.
No, I have not done it specifically, but I know that there are lots of papers where they do electron microscopy with this synaptosome.
Right. There was another question asking whether or not it was possible to do IP followed by the isolation.
Yeah, so there are people who want to get a more purified synaptosomal fraction. They do immunoprecipitation. They also do similar to FACS sorting. They call it FASS, so it's fluorescence activated synaptosomal sorting. So you tag your synaptosomal proteins with fluorescent proteins, and it's similar to your FACS workflow. Then you isolate it from your uh, supernatant or your lysate that you have. And people use that even for this technique where you have this layer and then you isolate only this layer of the synaptosomes, and then you purify it even further with fluorescent assistant sorting, or like you said, you can also do immunoprecipitation. So it is doable, and I haven't, once again, I've not done it with this protocol, but I know that it is.
Great, thanks. So the next question was asking when you do this isolation, are the synaptosomes intact or do they dissociate?
So at this point, they are intact, and I don't know if you can see it on the screen when I'm pointing. So it's in the step 3 where you have your crude synaptosomal pellet that is a fraction P2, and here it is intact. But once you use Triton X100 to separate, then it's not intact anymore. But then I would still say all your proteins are there, and I mean they don't degrade, so they do get enriched. But at this point, they do the Triton X100. It disrupts your membranes of your synaptic membrane, and then your cytosol gets separated. So at this point, they are not, but at the P2 level, they are intact.
Um, we also have another question asking specifically about your protocol. So it seems like this person had tried your protocol, but says my S3 fraction still contains as much chat as the S2 fraction. What can I do to improve the purity of each fraction?
So for that one, I think the most important thing is during the centrifugation steps. Sometimes your pellet may get disturbed and it could go into supernatant. So what you could do is you don't have to take all of the supernatant in these steps. You can leave some behind, and in this way you prevent disrupting the pellet. But once again, if your chat is there in the cholineacetyl transferase is involved in acetylcholine production, and there have been studies that show that this is also being present in the synapses. So you could have you also probably have to check your maturity level of your motor neurons. Maybe it's at a specific time point where you have more chat also being produced in the synapses so that you get faster acetylcholine production. This is an option. And maybe the protocol for generating neurons in your lab is probably different from ours. So it is attaining maturity even earlier, or maybe it's attaining it later. So it could be protocol dependent as well. So that's something you'll have to check probably by immunostaining or EM or even electrophysiology and have a look at how your cultures are. And maybe you can do like different time points, for instance, just to confirm if chat is always in the S3 in all the time points or if it's only in early time points or later time points. So that could probably be protocol dependent, also your disease or your model dependent as well. So uh, so what we have right here is specific, let's say, for our model and our iPSC-derived motor neuron protocol, and it could vary greatly with other people as well.
I think that's a really good segue to this actually, this question where someone was asking how long do the motor neurons have to mature in vitro in order to get synapses for the experiment?
Yeah. So this is something that we standardized in our lab. So our motor neurons, for the ALS motor neurons, for instance, they start degenerating around 60 days, 55, 60 days in vitro. So which means that it's too late a time point for us to do a study. So we decided to study them around day 42. That's around 6 weeks. And we start seeing synapses around 4.5 to 5 weeks in culture. So mature synapses. And we start seeing synaptogenesis in terms of like PCRs at around 3 to 4 weeks in our culture. So this is our protocol. So around day 21, we start seeing synaptic genes. Around day 30 to day 35, we see mature synapses in immunofluorescence. And we chose day 42 because it's like an in-between time point between mature synapses and degenerating synapses and ALS. And around 60, our synapses start degenerating in the ALS group. So once again, this is dependent on your protocol. I know lots of protocols where they can grow motor neurons up to day 120, for instance. And there are lots of disease models like spinal muscular atrophy, where you're looking at motor neurons already at day 20, an early onset disease, day 20, 25. But maybe the time is too early in your cultures to even have synapses at that point. So once again, this is something that you need to verify and see if it is relevant for your disease model and your protocol.
Kind of along that line, there was also another question asking, you know, if you have an experimental condition where you're trying to stimulate the motor neuron activity through, for example, KCL, um, is this possible with this protocol, especially given that stimulation must be short and that the protocol has some incubations?
Yeah, so that's going to be a bit tricky, but it can be done. For instance, when you do a treatment, there's going to be a brief incubation period for your chemicals to act, and then as soon as it's done, you immediately need to homogenize your neurons so that you don't lose out on the synaptic changes because synapses are very transient. They change and they're quite plastic. And some chemicals have an instantaneous effect, and some you need to incubate them for 24, 48 hours to see an effect, or some drugs, for instance. So you'll have to, once again, check the activity. If you're going to do electrophysiology, that's a very good readout to see how early your activity changes. And you can use that as your time point. Maybe it could be 5 minutes, it could be 1 hour, and then immediately you can start homogenizing the neurons. And once you add your protease inhibitors in this step and you do the homogenated, similar to doing a Western blot, the first step, for instance, and your whatever changes are there should be preserved. And you can, of course, check it later by blots and see if anything changes or not. And you can correlate it with electrophysiology or immunofluorescence or whatever technique you've been using to validate your drug treatment. So yeah, it is possible. It's just tricky and it's time sensitive. So of course that needs to be taken care of.
Great, thank you. I think we have time for maybe a couple more questions. So there was a question asking whether or not you've compared your protocol with commercially available kits in terms of yield and purity.
Yeah, so we have not compared them with kits, but I know that the kits they don't go. If I'm not wrong, I haven't checked myself, but they don't go up to the last fraction. And the kits, I think they stop at the synaptosomal fraction, if I'm not wrong. So it depends. I mean, in our case, the kits were a bit expensive, and I didn't want to invest lots of kits because we had multiple experiments planned, and we didn't want to invest so much money in buying so many kits. That's why we decided to have an in-house protocol that we knew it worked with rodent brains, so we just adapted it for iPSC neurons. But yeah, I mean, once again, the kits, I don't know if it has been validated for iPSC neurons. I know that the kits can be used for rodent brains or postmortem brains. So, but I'm not compared the efficiency between this technique and your and the kits technique. But of course, I mean, if you have funding and then if you don't need more of it, then obviously you can use the kit for your experiment. And I think the kits promise that there is a good separation. I think the kits are also based on a gradient, if I'm not wrong, but I don't know. I'll have to check. But yeah, I mean, if it all depends on your end users' requirements. So if you want to use the kit, you can.
Great, thank you. So in terms of, storage, what are your recommendations? So after you isolate your samples, do can you store them in -80 and for how long? Or do you recommend, for example, if you're going to do Western blot, preparing the lysate and storing that rather than the synaptosomes themselves?
Yeah. So that is a good question. So usually, since it's a short protocol, we do the entire steps in 1 day, and you can store all of these fractions at -80. So we usually snap freeze them really quickly, and then we just store them in -80, and then we've used samples that are like 8 months old as well that have been in the -80, and you can store them however long you store your lysates for Western blot. So it's a similar thing. It doesn't degrade over time. So what I would recommend is when you get all these fractions, you do multiple aliquots of them, smaller aliquots, so that you don't freeze thaw them multiple times. And if you're going to do proteomics, you all you have to do is like save some of the samples separately only for proteomics and never use it and use the rest of it only for Western blots. And if I remember correctly, you could stop at the step where you have the homogenate and you can freeze this. And then the next day you could continue with these steps. But then, like I said, since it's a quick protocol, we always did the entire steps in 1 day. And then we stored each of these fractions at -80 after aliquoting them into smaller volumes.
Great, that's really convenient that you can store them for a while.
Yeah.
So I'm going to ask one more question,
Mhm.
mainly because there was a few people that asked this. So is it possible to isolate perisynaptic astrocytes as well with this protocol?
That is a good question. So I know that there are protocols so there are lots of protocols where you isolate only the pre and the post-synaptic compartments, like this one. But unfortunately, in our protocol, you cannot get the astrocytic end feed. But I think those are called synaptoneurosomes. There are protocols where you isolate not just your pre and post-synaptic compartments, but also your astrocytic end feed and also microglial compartments that are involved in synaptic pruning. And that's a protocol that I haven't looked at much, but I have not used that. And I don't think with this protocol, I have not checked for astrocytic markers, but I'm pretty sure that we do not get those in this protocol. They get purified. You will probably see those in your S1 or S2 fractions, but you don't see them in your synaptic fractions. So I think it's a different protocol to isolate synaptoneurosomes that contain all of these.
Okay, great. Well, thank you so much, Sandeep. That was a wonderful, a great Q&A. Excellent questions from everyone. Before we end the webinar, I do just want to share a few more slides just to share some products that might be interesting for our audience today. So I'm going to go ahead and share my screen real quick. Okay, so at Protein Tech, we have a very large portfolio of antibodies that encompass two-thirds of the human proteome. This includes polyclonal, monoclonal, and recombinant antibodies against synaptic proteins. Most of our antibodies are validated in multiple applications, and our antibodies for key synaptic proteins are top-cited and highly rated by our customers. For example, on the right, you can see our PSD95 recombinant antibody and our synaptophysin polyclonal antibody both have an average 5-star review, and our synaptophysin polyclonal has been cited over 150 times. In addition to unconjugated antibodies, at Protein Tech, we also offer primary antibodies that are directly conjugated to correlite dyes for simplified immunofluorescence and flow cytometry workflows. So our in-house correlite dyes are bright fluorescent dyes that deliver high photostability and minimal fluorescent bleed-through, making it easy to combine 2 or more dyes in just one experiment. They have equivalent brightness to Alexa fluor dyes, and our antibodies are available in various conjugates, including 405, 488, 555, 594, 647, and 750. If you are looking for a conjugated antibody, but one is not available for you, Protein Tech also offers flexible kits for the direct labeling of primary antibodies. What makes these kits so great is that they are compatible with any primary antibody from any supplier, so it doesn't have to be a Protein Tech antibody. It also works with any antibody concentration and formulation. So oftentimes with more traditional methods of antibody labeling, you have to do some sort of buffer exchange to ensure that your primary antibody is in PBS only. However, with these kits, you don't have to worry about that. And so you don't end up losing a lot of antibody during that buffer exchange, which can end up being quite expensive. These kits also allow you to label just as much antibody as you need for an experiment. So again, with traditional methods of antibody labeling, oftentimes you have to label the entire tube, but here you can label just as little as 0.5 micrograms, or you can scale up if you need to. The protocol is really fast and easy. So within 10 minutes, you have a labeled antibody that you can use for your experiment. The way that it works is that it uses a high affinity flex linker. So you take your primary antibody, you introduce the flex linker, which has the conjugates covalently conjugated to them. And then within minutes, that flex linker is going to bind to the FC region of your primary antibody. And then you add a flex quencher to bind to any of the unbound flex linker, and that's it. And then you have your conjugated antibody ready to use. And because you're pre-conjugating your antibody, it allows for multiplexing with same species antibodies. Currently, these kits are validated in multiple applications such as IF, flow cytometry, and Western blot. And you can choose from flexible or flexible 2.0, which is our newly released flexible. And it has been optimized for IF. So basically, these flex linkers have more dyes. I also really quickly want to highlight our multi-Rab goat recombinant secondary antibodies. So our multi-Rabs are a multiclonal mixture of recombinant monoclonal antibodies, which recognize multiple epitopes on the same IgG. So the idea behind this was that we wanted to have a secondary that acted like a polyclonal, but had the superior specificity of monoclonals. And because these clones are hand selected, you do see very minimal cross-reactivity with off-target species. And because they are recombinant, they offer unlimited supply and high lot-to-lot consistency, making them ideal for your long-term projects. These secondaries are also validated in multiple applications, and currently we have these specificities and conjugates available on the top right. And then lastly, I just wanted to highlight some accessory products that we have available for Western blot, since I'm sure many people in our audience today are using this application. So in addition to over 20,000 antibodies that are validated for Western blot, at Protein Tech, we also offer loading control antibodies, including the highest cited GAP-DH antibody on the market. We also offer protein ladders that encompass regular, broad, and extra broad ranges. And we also have our in-house signal bright ECL substrates for the detection of proteins at femtogram levels. So with that, I just wanted to thank everyone for joining today. Thank you so much, Dr. Rajkumar, for the wonderful and informative talk. If you do have any questions, you can directly reach out to Dr. Rajkumar at sandeep.rajkumar@live.com, or you can email us at events@ptglab.com. If you liked today's webinar and want to stay up to date with any upcoming events that we have, please do follow us on our social media channels because we do post about all upcoming events there. So with that, I will go ahead and end the webinar, and thank you again, everyone, for joining, and have a great rest of your day.
Yeah, thank you as well. Goodbye.